Okay, so welcome to this next video in which we are talking about endocytosis, and where we're specifically looking at endocytic motifs. Okay, so we've now looked at the basic outline of endocytosis. What we're now going to look at for the rest of this video is this specific endocytic motif, okay? So we're going to look at it in a bit more detail. Right, so endocytic motifs then. Endocytic motifs. So let's say we have our target protein sitting in the plasma membrane of the cell, and we can imagine that it's either synaptobrevin 2 or synaptotagmin 1/2. So here's our plasma membrane here, and we now have some target protein sitting here. Okay, so this is our target protein. Okay, and we can imagine that it's synaptobrevin 2. We'll go for synaptobrevin 2. Partine, what am I doing? Target protein. Okay, right. And basically, this is the cytoplasmic domain of the protein. Okay, now in order for adapter proteins to recognize this cytoplasmic domain of the protein, what we need to have is certain combinations of amino acids in the polypeptide so that they can be recognized by the adapter protein. Okay, so in order for the adapter protein here, which we've always shown as this horseshoe so far, but we'll look at the adapter protein in more detail in upcoming videos. In order for this adapter protein to recognize that this target protein needs to be endocytosed, the protein to be endocytosed needs to have certain sequences of amino acids in it, and these sequences of amino acids which tell adapter proteins that this target protein needs to be endocytosed, that is what is known as an endocytic motif. So basically, within this cytoplasmic domain of this protein, so let me just label this up. This is the cytoplasm of the cell, okay, and this is the extracellular fluid over here, ECF for short. Okay, and on the cytoplasmic domain, which is just a fancy name for the portion of the protein that is in the cytoplasm, or on the cytoplasmic side of the plasma membrane, there will be a certain combination of amino acids which are next to each other, which will tell the adapter protein that this target protein needs to be endocytosed. Okay, and these are called endocytic motifs. So let me give you some examples of endocytic motifs. So basically, one of them is as follows. So you have an N, and I'm using the single letter amino acid code at the moment. So N stands for the amino acid asparagine, and I'll show you the structure of asparagine in a moment. So N is the single letter amino acid code for asparagine. Then you can have XX. Now, if you look at single letter amino acid code, there is no amino acid which means which we use X to denote. So why have I written X there? Well, basically, this means that you can have anything in these positions, okay? So after the asparagine amino acid, you can then have anything followed by anything, and then you need to have a tyrosine, a Y. So Y is the single letter amino acid code for tyrosine. Okay, so let me show you the structure of these two amino acids that are set, the asparagine and then the tyrosine. So, the asparagine is at the amino terminus here. So here's its amino group here, which will be linked to the polypeptide up here, okay? Then we have the alpha carbon of this asparagine residue with a hydrogen, okay? Then we have the carboxylic acid group here, which will be bound in a peptide link to the next amino acid along. Okay, now let's put the R group of asparagine. Now the R group of asparagine is that you have effectively an ethanoic acid group sticking off the side, but that ethanoic acid group has had an amide bond formed, basically, and it's been turned into a primary amide, which means that it has an amino group of this carbonyl group here instead of a hydroxyl group. So you've got a primary amide group here on the end. Okay, so that is the structure of the amino acid asparagine, rather. Okay, so now let's put the next one in, which can be anything. So I'll just denote this one's R group as R. So I'll just put R there, which just denotes that it can be anything. Okay, it just denotes the R group of the amino acid. So here's that next amino acid, so this is X. Okay, then another anything, so we'll have this again. Here's the amino group, here's the alpha carbon, and we'll have another R group, which can be R prime, so another anything. Okay, and then carbon R group here. Okay, and then after that, you have tyrosine. Now the structure of tyrosine is as follows. Here's the generic amino acid structure so far, the amino group bound in this peptide link here. Then we have our alpha carbon with a hydrogen off it. And then the R group of tyrosine is you have a methylene group, like so, with then a benzene ring off it here. Okay, so here's the benzene ring. And then off the fourth carbon of the benzene ring, you then have a hydroxyl group here. And then finally, tyrosine will end with its carboxyl group, which will be bound to the amino group of the next amino acid along in the polypeptide. So this is this endocytic motif. It's just a sequence of amino acids within the polypeptide of our target protein, which will be recognized by the adapter protein and can therefore tell the adapter protein that this target protein needs to be endocytosed. So this entire thing here, this is the endocytic motif. Again, let me just finish this by putting the Y there for the single letter amino acid code for tyrosine. So this is an endocytic motif. Okay, now it's not the only endocytic motif that you can have. There is another one that can be used. Okay, so this next one is slightly less specific than the one that we've just seen. So the next one is that you can have Y for tyrosine, okay, XX again for anything followed by anything, and then we put this Greek letter phi in there. So this is phi. Now what on earth does phi mean? Phi does not stand for any amino acid I know. Instead, what phi means is that it just needs to be a bulky hydrophobic amino acid. Okay, that you put in this position here. So what examples are there of bulky hydrophobic amino acids? Well, examples of bulky hydrophobic acids would be phenylalanine, which is denoted by the single letter amino acid code F. So phenylalanine would be an example of a bulky hydrophobic amino acid. And another example would be isoleucine, denoted by I. Okay, so I won't draw out the full structure of the endocytic motif again, but you can imagine we put tyrosine, which the structure of is here, we put that in position 1, we then have anything, and then in this fourth position here, what we put is a phenylalanine or an isoleucine. So I'll show you the structure of the phenylalanine or the isoleucine. So we'll put it as though it's in a polypeptide. So we'll start with phenylalanine. So here's the amino group as though it's in this endocytic motif bound to this third amino acid in the endocytic motif here. Okay, and then off the alpha carbon you have a hydrogen. And then effectively it's actually a very similar structure, R group, to tyrosine. You have a methylene group and then you have a benzene ring off that methylene group here, like so. And then you'll have this carbonyl group, which will be within the peptide link, so it'll be bound to the next amino acid along. So that is phenylalanine. So the important difference is that it does not have the hydroxyl group. That is why it becomes hydrophobic, because the hydroxyl group of tyrosine is polar. So that makes it favorable for this to interact with water. Without the hydroxyl group, suddenly you don't have any polarity here. These are very unpolar structures. Okay, so it becomes very unfavorable for this to interact with water, so it therefore is hydrophobic, it avoids water. Okay, right, so isoleucine next. So isoleucine structure, again drawn as though it were in a polypeptide. So here's the amino group, here's the alpha carbon, and the hydrogen coming off the alpha carbon. And then the structure of isoleucine is that you have these 3 carbons here, with a methyl group off this first carbon rather here, okay, and then hydrogens off all the other bonds available. Okay, so this is the structure of the amino acid isoleucine, single letter amino acid code I. Okay, so either of those two amino acids could be put in this fourth socket and you'd be fine, basically. They'd both be functional endocytic motifs. So those are two examples of endocytic motifs. So these basically are what the adapter protein is looking for. If you have these sequences that are exposed on your cytoplasmic domain, then the adapter protein will be able to bind to these sequences, and then you'll be able to continue the cascade of endocytosis. Okay, and we'll discuss a piece of an experimental corollary of this in the next video.